Just start by saying that Bats Without Borders, for those who don't know, is a small conservation organization working on the conservation of bats in Southern Africa through conservation, research, education, and capacity building. Now, Bats Without Borders is part of the bigger organization, Bat Conservation Africa, which works across the whole of Africa. And it's also part of the new global bat diversity network. Now, please join all these organizations and follow them on social media. So we're going to start the seminar, today's seminar, and let me just, excellent. So I'm really glad to introduce to you Dr. Teague O'Mara. Teague is an associate professor of biology at Southeastern Louisiana University in the US. He's a behavioral ecologist working on understanding how wild long-lived vertebrates integrate physiology and behavioral strategies in unpredictable environments. And he leads the Integrative Movement Lab, and this lab focuses on animal movement and physiology. I'm really excited to hear about what Teague has to say to us. So I'm going to stop sharing my screen. And I'll let you, Teague, share. And I'll just ask people again, if everyone can please make sure that they have their microphones off and muted and the videos off. Thank you very much. I'll move it on to Teague.
Thanks, Orly. It's great to talk to everybody today, and then I get to talk about one of the most conspicuous and charismatic bats in Africa, Eidolon helvum, straw-colored fruit bats. Today, what I'm going to try to talk about is we'll talk about some population level, sort of higher order level phenomenon with Eidolon, but then also we'll get into some of the nitty-gritty, well, not nitty-gritty, but we'll talk about how individuals move across the landscape and eventually come back to some ideas of how they connect to continents and why that's really important. Will you let me? No, there we go. Okay, now we can go forward. Excuse me. But I just want to start with that I am a very small part of this project. Team Eidolon at the Max Planck Institute of Animal Behavior is really coordinated by Dina Deckman, and we've had a huge number of people, including the students here that are highlighted in blue, that are contributing to understanding how these animals move and interact with their environments across various different countries in Africa. It's been a big project. It's been going on for a long time, and it's still ongoing with things like the Icarus project, where we'll be tracking migration. I'm in lots of different places, but also there's some other ways to get involved, particularly through colony counts through the Eidolon monitoring network, and I'll bring that up in just a little bit. But just a little perspective about how Eidolon, or what Eidolon actually is and where it sits within the bigger fruit bat phylogeny, the pteropodid phylogeny. Eidolon sits out on this very long and lonely branch. They're kind of an unusual genus. There's 2 species within the genus, but they've been essentially unchanged for a very long time, or at least their evolutionary history has been going on for a very long time. They sit next to a clade of mostly Australasian fruit bats, which is kind of interesting because they share some of the same kinds of behaviors. Particularly, we're going to talk about some of these migrating things that they actually do. And so Eidolon is going to give us a different kind of perspective about what it's like to be a fruit bat, particularly in Africa. So we like to refer to them a bit as a wandering gardener. They have an incredibly diverse diet where they're feeding on ripe fruit, nectar, bark, and leaves. These bats are sexually dimorphic. Females are a little bit smaller than males. But the really outstanding thing about them is that they roost in colonies that range from 100 bats, or maybe just a couple individuals by themselves, up to possibly 10 million bats. That's a lot of animals altogether. Eidolon is one of the most conspicuous and potentially numerous animals in the entire continent. Ooh. And they range across most of sub-Saharan Africa. They have an asynchronous reproductive pattern across the range, which is really quite interesting because we can look at the estimated start of the birth pulse in this little ring here, and colonies that are observed to having their major birth pulse are shown on the outside. And what we can see from that is that it's not necessarily geographically limited. It's not patterned very heavily, but some places have big birth pulses and others don't. Why this is, it's not totally clear. Because when we turn back and look at the number of males and females in a roost, so here roosts are shown geographically across the bottom, females in black and males in gray, the proportion of males and females isn't consistent across all of these roosts. So it could be that there are changing demographics. It could be that females are congregating into a maternity type colony. But again, the sources of those animals and where they come from isn't totally clear. What is also interesting to note from these captures of animals is that the proportion of adults in a roost is not consistent. So we have a large number of immatures right down here in Bioko, whereas we have almost completely adults in some of these other colonies. And the interactions between the birth pulses, the sexual dimorphism and differences between the numbers of males and females, and the proportions of adults hint at a potentially rich and complex fission fusion social organization. But it's not clear how that actually happens, because how do you measure what a social group is, what social associations are, how consistent they are across time in groups and colonies of potentially hundreds of thousands of animals? And like I said, eidolon is found across sub-Saharan Africa. What I'm highlighting here are iNaturalist records in orange, in the orange boxes, but then also where eidolon have been actively counted by the Eidolon Monitoring Network across primarily West and Eastern Africa. And we can see that there are places where there are really hotspots of eidolon, and they are moving in and out of these regions. Animals are not in the same roost all the time. Roosts are seasonally occupied and abandoned, but they keep coming back to the same places over and over and over again. And what we can see with some of the counting data is that we can highlight all of these different places where people have been counting bats, and the maximum colony size is highlighted by red, or by the colors from blue to red, from a little bit number of bats to a lot of bats. And the number of animals across a year are going to be shown by the size of the circles. And what we can see is that, hopefully we'll see, there we go. This is going to change across a year. So they're going to flash up and they're going to flash out. Some, for example, in Kasanka in Zambia, animals are only there for 2 or 3 months a year. But in Accra, there are consistently animals around. In Rwanda, there are consistently animals around. And why this actually occurs is not totally clear. We think it's probably tied to food availability, but how did they anticipate that and how do they predict it? It's not really well known. And the people who are figuring out how this actually is working is the Eidolon Monitoring Network. And this has been a network of people who are volunteering and working to understand how the demographics and how the population sizes change across the year. And these are done through standardized regular counts of eidolon roosts. Going out, you can count the number of bats in a cluster, the number of clusters on a branch, branches on a tree, and kind of scale up to figure out how many animals are around you. If you are living near one of these roosts and you're interested in joining up with the Eidolon Monitoring Network, Natalie has probably already gotten in touch with you, but if you would like to participate and learn more, please let me know. I'll put you in touch with Natalie Weber. I didn't feel comfortable putting her contact info up here without asking her right away. So I'm happy to leapfrog you into Natalie's welcoming arms. But these animals are all over the place. They're moving from one roost to the next. They're occupying and abandoning roosts, but why do they have to move so much? And we think a lot of this comes back to the differences in vegetation across the year. So the differences in food availability. So NDVI is a measure of greenness. It's a measure, it's a satellite-based measure of greenness. And we can see that, for example, in the savanna zone, there's relatively little greenness that's happening in March, but this becomes very nice and wet and green in September. So it could be that these bats are tracking this proportion of greenness and food availability across a year. And if we look at population fluctuations in gray, these are normalized to the maximum number of bats that are there in that given year. And we look at the changes in greenness in green, we can see that there are some sort of patterns that start to fall out a little bit. First off, you can see that in these 3 chosen colonies, bats arrive and they leave very quickly. So there are very steep changes in population size. Why that happens, it's not really clear. But you can also see that there are nice fluctuations in greenness. So it could be that these bats are tracking this. We're not really sure yet how that actually works. And this is a problem that a postdoc, Edward Hermy, is going to be tackling at Max Planck right now. But while we can start to sort of exchange or figure out why these animals are moving from one to the next, it doesn't really explain why there are hundreds of thousands of bats in Accra and maybe 8 to 10 million in Kasanka and 1,000 in other roosts on their own. And it kind of comes back to a question of how do you actually count a lot of these different kinds of things? Like, how do you actually count a colony that comprises 2 million to 10 million bats? When you have so many animals flying around and they're not roosting in a place that's easily accessible, it just becomes sort of a daunting task of trying to figure out how many bats there are around here. And we've taken a different kind of approach to use photogrammetry and computer vision to figure out, in particular, what the colony size and fluctuations in size are at Kasanka National Park. This is one of the largest roosts of Eidolon, if not the largest roost of Eidolon that we've ever measured. It may be possibly, if there are 10 million bats there, the biggest congregation of mammals anywhere in the world. And all of these bats roost in an tiny little forest that is 1 kilometer long by about 200 meters wide. So it's tiny. Except during the day, when you look over that forest, you cannot see a single bat. If you crawl underneath it, then you can. It's relatively low. And so what we've done, we've stationed GoPro cameras around the perimeter of forest to record them as they emerge every night. And so by doing this night after night after night, we can zero in on an actual number of how many bats that are there. And we can estimate and get a little bit better of an estimate of what's going on. Because in the past, this has been done by eye, which is a great first start. And it might, who knows, be more accurate, but we don't actually know that. We don't actually have any good measures to parameterize this. So we've been trying this with computer vision. And this is basically what it looks like, is that we track each of these little dots across the screen and then count them as they are going to appear crossing that red line. And so you can see this happening here. It's a little bit mesmerizing and kind of calming, just sit and stare at little dots flashing. But we can track the animals as they go across the screen. If they come back going downward, then we can subtract them away. But we can do this over and over and over again. It's a readily scalable technique. And this is being led by grad student Ben Koger at Max Planck. So we've been talking a little bit about what happens if we study these animals or count them at a population or a colony level. And this tells us a lot about what actually is happening with them. We see that these bats move around a lot. They're moving from colony to colony. The numbers are changing quite a lot. But the patterns of colony level movement are not necessarily straightforward. They are likely tied to reproduction somehow. And they are likely tied to resource availability. But we don't have great information about what actually happens just by looking at these population level metrics. So we have to then turn to tracking actual individuals over time to get a real understanding of how individuals occupy space within a colony and how that colony then occupies space within a landscape. And so we can build individual level data, particularly using tracking technology like radio collars or GPS. I'll be talking today mostly about GPS. So that we can get back to this idea of how bats influence a landscape. We can use individual level stories to build up how individuals, again, affect a group and affect a landscape. This will also let us know what a colony actually means. Eidolon are incredibly difficult to capture. They're incredibly difficult or almost impossible to recapture. So it's hard without these individual level tracking techniques to understand how individuals come and go and interact with each other. Because overall, we should expect that colony size influences movement of these animals. Because if you have 2 million bats who are your closest neighbors, that should be influencing your foraging competition as well. Along these lines, we can then come back to understand how foraging success and food availability influence the decision to migrate or not? Or is it just decided by simple social interactions? We don't actually know this. But overall, I think that we can take individual level stories of Eidolon Helvum to connect continental Africa in a variety of ways and understand what those consequences of those individuals moving across the landscape actually are. And so Max Planck has been using a variety of different kind of tracking technologies that are GPS based to understand how individuals move throughout a landscape. And today I'm going to be talking about a variety of those different kind of techniques and technologies. Actually, I'm not talking about technologies, just the results of the studies. But I just want to highlight that all of the tracking data that I'm talking about today are available on the MoveBank data repository for you to download, play around with, understand a little bit more about Eidolon if you so choose. So one of the things that we know about fruit bats in general, and Eidolon in particular, is that they are essential seed dispersal agents. Bats have the capacity to poop while in flight. They can disperse seeds into unoccupied aspects of the landscape, and they can disperse seeds further than other terrestrial species. They're not seed predators in general. If they do swallow seeds, they're coming through whole, and they're coming through with a nice little packet of fertilizer as well. So we can use the actual movement of animals, of individuals. We can see some animals that are tracked in Accra and in Chibi, and we can see that these animals are repeatedly going out to the same place over and over and over again. So we can use the geometry of that movement track. We can use gut passage times of actual dietary items. These were collected by Michael Abedi-Larty when he was a PhD student. And we can integrate those with the counts of the landscape from the Eidolon monitoring network to estimate how Eidolon affects its overall landscape and its seed dispersal services. What we can see just to start out with is that based on how long they keep seeds in their gut and how far they actually move throughout a given night, we have a variety of different kinds of seed dispersal potential distances. And this is going to change whether or not there's a lot of animals shown here in red, or there are relatively few animals shown in blue. This is going to go up a little bit and down. But there are some very long tails for how far seeds can be distributed. And we know that Eidolon seed dispersal scales with population size, because this should be a simple effect that you add more bats feeding, you add more seed dispersal. In the dry season in Accra, and in particular, is when we have the most animals that are around there. In the wet season, there are relatively fewer. But overall, they're putting out a sort of linear effect of seeds into the landscape. But what we can do with the movement data is really see how they move away from their central roost in Accra, for example, and push out into the landscape. What is the likelihood they're going to be putting seeds out into a landscape? How often is it going to happen? And is it going to be happening in the same place over and over and over again? Dispersing seeds right around one single roost doesn't do you much good. You need to, from a tree's perspective, it needs to go out further into the landscape. And what's, I think, really fantastic from this study that Marielle led is that we can take overall deforestation in a single environment in Ghana from the period of like 2001 to 2016. This has affected about 128,000 hectares. That's caused a decline in gross revenue by 133 million US dollars. But if we match up satellite images of those deforestation events over time and also our seed dispersal map, we can figure out what's the likelihood of bats actually taking seeds into those deforested areas. And we can see that during the peak colony size in both Accra and Chibi, that they're dispersing in between 200 and 800,000, or sorry, 200, 800 seeds into a huge number of hectares of land. So they're potentially reforesting quite a bit. And the economic value through things like stumpage fees and others could be upwards of $800,000. So not only is Eidolon playing a key role in the dispersal of seeds and maintaining the ecosystem, but they have a key economic role as well. But we know that these colonies are changing size. How does the changing size actually influence movement? And so we can look back at the Accra colony as they go from 150 to 100,000 150,000 to 1,000 animals, and we can estimate that, or we can predict that changes in population size of this magnitude is going to influence competition and how you forage. So you should be foraging further, longer, and potentially spend longer time in a patch if you have a lot of competitors. And by GPS tracking males, which at that time they were the only animals big enough to carry the GPS tags that we had, we can figure out how this actually is structured. Because what we see in small colonies, or when there's times of relatively few animals, they actually don't fly quite as far as when there are a lot of animals, which seems to suggest that colony size is influencing foraging movements. And overall, we see that their foraging distances over here on the left, in the wet season, they're not flying nearly as far as in the dry season, when there are many more bats. And they all are flying to different kinds of areas. In the wet season, there is less tree cover than in the dry season. So it seems that there are shifts in the ways that these animals are using their landscape and using their environment based on seasonality and population size. But there's one small complicating factor with this, is that in the dry season, when they're flying further, they're eating nectar, they're feeding on flowers. So they're flying very, very far to find this particular resource. Whereas in the wet season, they feed primarily on fruit. So we sort of have to start taking a more comparative approach to understand maybe how does the resource itself and not just the population size influence movement of these animals. And so this is a study that was led by a master's student, Camila Calderon, where she took a lot of the tracking data that was collected by the Institute from 4 sites in West and South Africa, or Southern Africa, from times when there were lots of bats and times where there were relatively few bats, to understand really what happens with their movement. How are they going about their day? And we can see this, for example, in Ghana, in Ouagadougou, and then also in Zambia, in Kasanka National Park. And these are all going to vary by population size. And so we can come back and make predictions about what we'd expect from theory about how eidolons should be foraging in a landscape. We generally should expect that foraging distance will increase with colony size, especially if we go back to those Accra data. We don't know how that should scale, because it's not really clear how things should scale from 1,000 to 8 million bats. But in general, we expect an increase. We also expect that as colony sizes start declining, bats should increase the number of foraging sites that they're using, because they're either scouting or the quality of those foraging patches is probably going down. So we should start to see some interactions between the colony level demographics and the ways that the bats are moving. But overall, what we've ended up seeing is that colony size has very little impact on the foraging patterns of these animals. The standout is going to be, again, back in Accra in the dry season, when they're foraging on flowers. The rest of these animals mostly seem to be foraging on fruits. And so it could be that nectar is driving these really outstanding distances, and we're starting to get evidence from other kinds of bats that seems to be the case, that nectar is sort of an unusual resource and really promotes different kinds of behaviors in bats. But we see a relatively consistent number of foraging sites that are used per night. The total distance traveled per night doesn't really change very much, which kind of gives us an idea of what standard eidolon behavior might actually be from this aggregation of multiple individuals. What we can see overall from the flight data is that their sort of overall speeds in terms of commuting time versus distance traveled doesn't really change very much across the night. It scales the same no matter how far they're actually going. Our largest colonies, though, in Kasanka and in Accra, are flying the farthest distances. So while their means may be relatively similar, particularly with Kasanka, with this big blob of bats down here, they are flying pretty far in some instances, which is an interesting thing. But what I think is also quite cool, or who knows what actually it means, is that the maximum here seems to be consistent across these 2 big colonies. And maybe that we're actually reaching the maximum distance that a bat can fly in a single night. So how do bats maintain this sort of conserved foraging pattern? It seems like eidolon forage like eidolon do, which gives us a little bit more of a hint that once eidolon cannot forage in the way that they normally are used to, then they just migrate and move to a new site. But from some work that we've done and also with near Sapir, we see that bats modify their flight with wind support. It's what flying animals should do. That tailwinds, when you have lots of wind at your back, should increase your ground speed, but you don't actually have to fly as hard. Your airspeed can decrease. And when we look across all of those different tracking studies, we see the same thing happening. So that when there are headwinds, animals speed up their airspeed, and when there are tailwinds, they slow down. And as they move across the landscape, their ground speed, so the speed relative to a fixed point on the ground, increases as they have tailwind support. That's what we should be doing. But it doesn't tell us how they actually make that happen. And so we can turn to another set of integrated technology into GPS and biologging trackers, an accelerometer. And that's going to let us measure changes in movement along 3 axes at between 10 and 100,000 times per second. What an accelerometer can do is it can quantify the actual movement. It doesn't really cost a lot in terms of battery power. GPS is a very hungry device. Accelerometry isn't, but it does generate a lot of data if you're recording 100 times every second. And what this can do is it captures animal motion. So we can see, for example, with this eidolon flying, we can see every single wing beat that this animal is executing. It would be nice then if we could actually see how they tilt their wings and how they do things like that, but that's beyond the ken of this study for right now. But we can use movement as a movement from acceleration as a proxy for energy expenditure. And this is done primarily through dynamic body acceleration. And because movement is the greatest amount of energy that we use every single day, sitting here, I'm not spending much energy. If I go out, walk and run, my energy expenditure goes up quite a bit more. But if we sum up the total net movement across all the axes, we can get a measure called ADBA. And what we've seen in other species, as they increase their speed here on the x-axis, they increase their ADBA. So their speed is increasing with this great cormorant linearly. With this border collie, we see that there's a sort of saturation eventually at the top. That might be a function of the device itself. But ADBA is measuring energy expenditure in some way. And so what we can actually do is take the accelerometer, extract out flight behavior, so how they're beating their wings. We can get ground speeds and air speeds, and understand a little bit more about what energy expenditure is like. Because we can also make some predictions about what we should see from accelerometry signals in bats. In general, as they beat their wings faster as they beat their wings faster, they should be accumulating more ADBA. They maybe are beating them harder. As they get that wind at their back, that wind support, they should have to work less hard, so their ADBA should go down. They're not pushing very hard. And we should see a curvilinear relationship with their airspeed. And this is based on some aerodynamic theory and the ways that other animals fly, is that when you fly very slow, it costs a lot of energy, and it's really, really hard to fly slowly. That's why we have so few animals that actually hover. As you fly very fast, you're generating lots of drag on your body, so that also costs a lot of energy. And so we can use insights from the movement of the animals to understand how their energy expenditure might change over time as well. And this is going to come back to how their foraging, how successful their foraging needs to be, and how constrained they actually are in their movements. And overall, what we see is that accelerometry and ADBA capture bat flight pretty well. They meet our expectations for how ADBA is accumulated with wing beat frequency. It goes up. As they get more wind support, it goes down. ADBA goes down, so that's good. That's what we expect. The one little tricky bit, which is sort of one of our best measures of power output and energy expenditure in bats, is what should be happening with airspeed. And we find a general decrease in ADBA with their airspeed. And this is probably a consequence of the way that bats fly. When they fly faster, they don't beat their wings faster like birds do. They don't really beat their wings so much harder, but they change the shape of their wings and they change the kinematics of their wings. And so by altering the way that their wings are sort of functioning as they fly, that might be generating less ADBA. So it's not a great measure for us right now. but it does give us an understanding a little bit more about what actually is happening with these bats. And it gives us a launching point to understand things a little bit more clearly further. And so from some of these local tracking studies, what we can now see is that Eidolon, they reuse the same feeding site many times. This is what people have seen with other fruit bats and other frugivores for quite a long time, is that once you find a fruit tree, you go back to it over and over and over again. It just could be that for Eidolon, that fruit tree is 30 kilometers away. So this hints at exceptional spatial memory and spatial maps of their landscape. This has been really well documented now for Egyptian fruit bats, for rosettas, that they have really exquisite understanding of the way that their environment is shaped and their place in the environment at any given time. What we know is that Eidolon forages in the same way across all colonies and regions. This is really an important little piece of information because it lets us take the movement of animals, it lets us take that information about seed dispersal that we looked at earlier, and we can actually then now input that on every single place where Eidolon is, because they're foraging the same, basically foraging the same way all of the time. This is a really powerful tool for us to understand the importance of Eidolon in a given region, to communicate not only its ecological, but its economic importance, and sort of come back to some of those human dimensions of bat biology and bat behavior that were talked about in the last Winged Wednesday webinar. So this is a really interesting place where we can start understanding a little bit more about how individual level stories, colony and sort of group level numbers influence sort of their role within a system itself. But yet we still don't understand yet how migration contributes to these population dynamics, because as animals are moving back and forth through all these different kinds of areas, my bats are the same as your bats and your bats are somebody else's. So it really ties back that we have to put all these animals together in a single population to understand their management overall. So this comes back to the last biggest question that I'm going to talk about today, and that's about Eidolon migration. And unfortunately, it is the least satisfying one right now, because we could talk about a couple of things that we know about Eidolon migration, but it is a lot that we don't. These are, despite being one of the most conspicuous animals, they are one of the most elusive in terms of their migration. There was some satellite tracking that was done in Zambia by Heidi Richter. And what we can see from those data is that animals mostly move into the DR Congo. We'll see a map of that in a second. And they fly about 90 kilometers a day. So they're flying around here. So 90 kilometers a day is well within the foraging ranges of our animals. So for them, a migratory step isn't that big of a deal. And so far, most of the migration work has been focused on Kasanka because it is such a huge sink of animals. There are millions and millions of animals. So they must be coming from a large geographic region. They must be. We just don't know where those geographic regions really are. Through our tracking and through radio tracking, we see an interchange between populations in Accra. So bats will move from Accra to Chibi and then move back and forth. Those are relatively close by. But we haven't really seen what the migration patterns for these areas really are yet. And so here are the data that I replotted from the Richter and Cummings study. And here is Kasanka in Zambia. And most of the bats are moving in this sort of, I guess, northwestern axis up into DR Congo, which is pretty interesting, like that they're all sort of going in this way. Does this mean that all of the animals from the greater Congo basin are draining into Zambia for that one sort of 2-month period during the year? We've been trying to do some other sorts of tracking from Kasanka, and we're seeing similar movements. So we're seeing similar movements up through here. We're seeing similar movements up into the Great Lakes region, but they're also moving directly to the east into Malawi. We have a little bit of movement to the south. What we don't actually see is bats moving this direction yet. But again, there are millions of animals at Kasanka. We're only able to tag a handful at most any given year. We would like to tag 100, but even then, that's a tiny, tiny, tiny drop in a bucket when there are potentially 8-10 million animals around. What we do think we know about Eidolon is that they follow the fruit. This is, again, data from Heidi Richter. And we can see that these are the number of trees that are bearing fruit for various fruit species. And we can see an increase overall in October that's going to peak somewhere around early November. And this is actually the box that shows when bats are actually there. So bats are arriving somewhere mid to late October. They're peaking late November, and then they're leaving by the new year. So bats seem to be at least coming into Kasanka when there's fruit. We see similar things from Accra and Chibi in Ghana, or these are counts of bats shown in light and dark gray, and here are phenological estimates along the lines. And what we can particularly note here is that as phenology increases, so does the number of bats. We're going to wrap around back to the end here, but we'll eventually reach a phenological peak and bats are going to drop off to a low level. What's really not quite clear is from Chibi, is that we get our peak here when there's relatively even levels of fruit availability. So it seems like bats are following the fruit, but still, how do bats connect different areas? And how do they actually anticipate the fruiting phenology across these really wide geographic regions? So one of the last things is, what do we actually need to find out about eidolon migration? So my first question really is that, do they actually migrate? Do they come back to the same places over and over and over again, like we think with traditional migration? Or do they wander across a range following fruit? This would be something much more similar to the Chiropus species in Australia. Also, how do colonies decide when they're actually going to move? So we saw in those colony counts that are normalized to a maximum population size, that they're really sharp. They go up and they go down. How does that actually happen? That all of a sudden everybody decides to go. There are some hypotheses about how and why that actually could happen, but those are going to be very difficult to test in the future. Also, how are colonies linked to one another? Are there migratory routes? Do we have divisions in the continental range? So do we have, for example, a sort of northern migratory route and a southern migratory route that eventually come together somewhere in equatorial Africa? Because this is a panmictic population where we don't see genetic differentiation across all of the sampled animals that have been genotyped in their cytochrome B, it's not super clear exactly how that can happen without animals moving all over the place. But this is going to be tied back to how migration and reproduction are linked back to one another. Because we had these sort of birth peaks all over the place that are not clearly geographically delineated, there's something much more interesting that's happening in there. And lastly, how do they actually really connect the continents? Despite being one of the most conspicuous and abundant animals, we don't really understand how it moves. What we do know through the colony counts in the eidolon monitoring network is that populations are declining for a variety of reasons. These bats are particularly susceptible to hunting because of their low reproductive output. And this is the same for all bats. It's just that these are really big pieces of animal that are running around there. And populations are not really able to rapidly recover when they're heavily hunted. But these animals are also nuisances. They roost where people are living. And so there, in some places, are strong pushes to remove their roosting trees. And this also has a dramatic effect on their population stability and their overall numbers. But this sort of brings up another issue of, if these animals have such a strong ecosystem services effect, how does removing them through these ways shift their services across a range? So if a site completely loses eidolon, it will lose out on all the ecosystem services they provide. And we're seeing this with like declines in insectivorous bats, but these fruit-eating bats are essential seed dispersers for their local ecosystems. And the last thing is, zoonosis is a real effect from these fruit bats. And we still yet don't really understand how their movement is tied back to reproduction, is tied back to potential zoonotic outbreaks. But it's really something that is sort of ripe for understanding a little bit more clearly. But because these animals are such essential, play such an essential role in their ecosystem, it's really essential to understand sort of how they, as an ecological species, interacts with us as a human species. So lastly, I just want to leave with one last photo from Christian Siegler that just kind of shows the massive number of animals that we're dealing with in any given one place at one time. Eidolon helvum is sort of a spectacular animal that is ripe for answering lots of different kinds of questions. But some of the most interesting questions, I think, for them are going to be the most challenging ones. And we're going to be addressing those through various different kind of technological solutions in the future, but it still takes a lot of on-the-ground work and cooperation from lots of different partners that help us understand what actually happens with Eidolon overall. But that's it. That's all I have to talk to you guys about. I'm happy to answer questions as best as I can. So please just let me know what you want to talk about.
Many thanks, Tig, for such a great talk on such a really iconic bat species. If anyone has any questions, please write them in the chat, or you can raise your hand and I'll call your name out. Okay, we got somebody, Dr. E. Alberts, waving their hand. If you unmute yourself.
You have to unmute yourself. Oh, I can't hear you.
So maybe while we're waiting, I'll ask you a quick question.
Oh, sure.
I just was wondering, oh yeah, we can hear now.
Okay.
No. Yeah, so I was just wondering, you were talking a lot about an energy expenditure and how it affects their flight and the distances that they fly. Yeah. I was wondering, how would this be affected by climate change?
That is an excellent question. I think the biggest effect that we're going to see with climate change is that they're going to have to be either moving further or less far depending on your models. I think what we'll see with climate change is that their ranges will get constricted more, especially with drying effects, and they're really going to upend some of these phenological patterns. So what I think the biggest effect is actually going to be is that places are going to lose out on Eidolon. Their energy expenditure won't change much because it seems like Eidolon do what Eidolon do. And they go out, they fly, they forage, they meet their needs. And when their needs can't be met, then they move on. If they find those places where they can no longer be met, then we will lose Eidolon. But I think that most likely it's that their range is going to be highly constricted, which is different than lots of other species that are starting to expand their ranges.
Thanks, that's really interesting. Obviously, climate change is my interest, so that's why I'm asking. We have some questions here. So I have a question from Francisco Amorim. He's saying, sorry, he's in the office, he can't use the mic.
Oh,
I'll read it out then. You mentioned they eat fruit in the wet season and nectar in the dry, but this was mostly the case of Accra colony. Is that correct? Is that matter of availability or different energetic requirements? Does these different strategies reflect in the number of individuals in the roost?
So I'll take that last question first. These different strategies, we don't actually know if they reflect the number of roosts because they're so highly correlated with one another. What we do know is that they are feeding on flowers because flowers are most often available in the wet season or the dry season, sorry. And so that's when they actually have the chance to go out and feed on them. Why that is, it's not super clear because nectar is not a super abundant, or sorry, a super energy dense resource, but it could have lots of different supplemental minerals and things like that that are in it. So it's not clear why they go so crazy about it. It's something that I'm super interested in. Like, why do some bats just fly super long distances to feed on something that is not a main part of their diet? We don't, Accra is where these bats have been best studied. So we don't really know what happens in other sites very well. We have some feeding data from other locations, but Accra is really where they've been best studied for that kind of information.
Okay, thank you. And we have another question from Marie Hanley asking, have you seen white nose syndrome in Eidolon?
Not that anyone has ever reported. I wouldn't expect it to be found in Eidolon just because there's such a tropical species. And white nose tends to bottom out once it starts to get a little bit warm. Like, I live in Louisiana. We're surrounded by states, which is in the southeastern part of the US, the Gulf Coast. And we see white nose appearing in places like around here, but in my very hot and very humid location, we're luckily not seeing that quite yet.
Great, thanks. So we had a question about whether it's going to be uploaded, and yes, we're going to upload everything into the YouTube channel. So if anyone missed anything, you'll be able to hear it then. Another question we had from Rina Ullman saying, what a wonderful talk. Do the straw-colored share food with each other the same way that vampire bats share food?
Not that we know of. It's probably likely that moms share food with their kids. Like they're probably sequentially provision them with fruit in some ways, but they're not going to regurgitate it and do things like vampire bats. Most of the frugivores, when they're adults, they're pretty selfish about handling their own food and eating it. They might try to steal it from each other, but they're not as generous in sharing as vampire bats.
Thanks. And Javier José is saying, great talk. Have you tracked any offshore movement? Any explanation for them?
We haven't seen any real offshore movements yet. I'm sure if you go back and track animals in some of the islands, you might see some offshore movements, but they might be a function of commuting around areas. So I haven't seen those yet with these bats.
Okay, thanks. And we have Julian Capel asking, do you know if the increase of greenness measured by NDVI is associated with flowering of fruit production in your study sites?
Thanks. So I don't know this for certain for all of them. There's so many study sites across a very wide geographic region, but in general, NDVI is much more closely associated with fruiting production later on. So lots of species, there are always exceptions to this, but a lot of tree species will prefer to, well, they flower when it's dry and they fruit when it's wet. And this is just a function of being able to disperse pollen.
Thanks. So we have a couple of questions from Bahirathan, and I apologize for mispronouncing people's names. The first is, did you find any age effect among the tracked males? Do younger males travel or commute further than older ones?
So we don't see that quite with our data, but we weren't able to age our animals outside of sort of reproductive status. So are they scrotal or are they not? So the aging techniques that have been done at Eidolon are done from tooth rings, so tooth annuli. And so you have to extract a tooth out, and we haven't done that because it's much more intensive to do that kind of stuff. But it's cool that you might be seeing that with flying foxes in India. I would really be interested to see those kinds of demographic effects on their movement. And we do have plans to tag females in the future. We have been tagging females, and we have tagged them in the past, but they're big girls, so they're big females. And so for a lot of our studies, just to make them as a sort of coherent data set as possible, we only include the male data in some of the tracking. But for example, there are females in that data set that I shared with you guys earlier on.
Thanks. We got a question here, which is actually pretty much for everyone, but I suppose mainly asking you. I would like to ask an advice. This is from Francesca Cosentino. I'm going to build an African plants richness map, bat food items, with macroecological approaches and by using citizen science data. Do you think this map could be a reliable proxy of trophic resource availability for frugivorous bats in Africa?
It could be, but we have to also know what they're eating. For lots of species, we don't have a great idea. They could be pretty diverse. There is a database, a plant food database for bats, and I can look that up and share it with you guys too. But like any kinds of information where we have any kind of richest maps that we can put into things like map of life, or correlate with our already known food lists is always super helpful.
Okay, thanks. It's definitely going to be a useful resource, I'm sure, for a lot of people.
Yeah.
We had a question from Fiona Matthews asking, is anyone quantifying fruit crop consumption by the species and comparing it with benefits from pollination? The case for forest regeneration is clear, but those benefits may not be of such interest to farmers directly impacted by fruit consumption.
Yeah, that's a tough one. So the good thing is that these bats are also pollinating those fruit crops. So you have to take the good and the bad. I'm not totally sure if anybody is trying to do this off the top of my head. There probably is somebody. The conflict of eidolon and people is, I mean, it's true, it's there and it's true. And I could think of like Paul Vibala in Kenya has been doing this kind of work to discuss how people view the animals, like what their biggest problems are with them. And oftentimes, at least from what I've read in Paul's work, it's not the fruit crop consumption that's the biggest problem. It's the roosts that are the biggest problem for people.
And I'm guessing they roost mainly in trees, aren't they?
They roost in very tall trees. They like big trees. And so that's why, like, if there's a village with a big tree in it, it's usually often in the center of the village where people are gathering, and that's where the eidolon like to be too.
Yeah.
Yeah.
So it's the usual conflicts between bats and humans.
Yep.
We have a question by Oliver Lindeck, who's saying, crying baby on my lap. So, but it still has a question. Are there any paleontological findings indicating much larger ranges or other now abandoned colony locations in other parts of Africa?
Whew. We generally have really terrible paleontological record for bats. They don't fossilize very well. We have really, really, really old bats. We have some younger bats, particularly like in the Caribbean, but we don't have great data. Like any paleontological findings from tropical species is a big deal because things don't fossilize very well in the tropics. So we don't have great data of abandoned colonies, but we do have historical records of many of these colonies, particularly I'm thinking of Accra in general, that have been there for hundreds of years. So these are really persistent colonies that people have observed and the bats have been using.
Thanks. And then we have a question from Teresa Laverty saying, great talk, Teague. Curious if you have any insights why eidolon is occasionally observed in the Namib Desert on the Atlantic coast of Namibia. We also assumed these were individuals that were blown off track with the east winds in winter, but not sure if you have any other ideas.
Nope. Unsatisfying. They're probably blown off course. They could be exploring, but the Namib is a very strange place. I would not expect them. Well, it's not a strange place. It's a wonderful place. I've never been there, but it seems beautiful. But eidolon, I wouldn't expect there. We do see eidolon in the Arabian Peninsula as well along the Red Sea. But those are locations that there are a lot of fruit available for them. And Kara Rogers replies that the database I was talking about with the plant foods and bats is batbase.org. So that's available for everybody too.
Excellent. We have a question from Julio and Erna saying, thanks very much for sharing, Teague. Have enjoyed it. Was wondering, what was your estimate of the Kasanka population? You may have mentioned it, but I was stealthily watching from work and unable to give attention.
I didn't give an estimate because we don't know yet. It's still we're still refining things a little bit to get a good count, because when we do have an estimate, we want to make sure that it's a really good one. So that will be coming up in the future.
Okay, thanks. And a question from Julian Capel saying, we took some footage of the Yaoundé eidolon colony at dusk when they leave the colony. Do you think the tool you're developing for automatic sensors could apply to any colony?
Yep, I do. I think that it can be a broadly deployed tool. And once we actually get it finalized and set and we're happy with it, we'll share it with everybody.
Excellent. And the other question was, are you interested in the footage we took? And I suppose you can discuss it.
We can discuss that later. Yeah, maybe send me an email. We can talk about it because we'd be interested maybe in getting multiple cameras. So if you have footage from lots of different spots around there, so we could get a better estimate too. But any estimates for like Yaoundé would be really wonderful. Yeah.
Yeah, excellent. So we don't have any more questions, but I'm quite interested in just asking you again about relating some things to the genetics work you mentioned.
Yeah.
And you said that they're all part of one panmictic population.
So it seems. This is all based on cytochrome B work.
Yeah. It'd be quite interesting if they did a bit more fine scale work using genomics, for example, and seeing whether you see any functional differences, maybe even adaptive variation that varies between these bats.
Yeah, so these are plans that are in the works. Because we would really, really like to understand this a little bit more. I mean, cytochrome B was a great tool. It still is a great tool, but it doesn't get at the fine scale differences that you're talking about. hopefully that's going to happen relatively in the near future. Yeah.
Brilliant. I look forward to hearing about it. We have some more questions. We have only a few more minutes left, so I'm going to read a couple more questions. Bennett or Obite asked, apology if your talk already touched on this, but I was going to ask if you know why some individuals stay behind while migrants leave.
Nope. That's one of the things that we would really, really like to know. Because also what we're seeing are getting hints at from other places where there are permanent colonies, like we have Rwanda Wildlife Conservation Association in Rwanda has been tracking bats for a while, and it seems like the population stay the same, but animals come in and they come out. So we don't really know even if they if they are the same animals all the time. There's so much more to learn.
Yeah, it's great when you do loads of work on a species and you're just touching the tip of the iceberg. And
yeah, it's awesome and frustrating.
Yes. It's many more years of researching it, that's for sure. Okay, so I can't see any more questions coming, but many people have mentioned that they really enjoyed the talk, and thank you for that. So I would like if everyone can turn their microphones on and we can have a bit of a clap of hand to thank Teague for the great talk.
Thanks everybody, it was super fun. These are great questions.
Thank you. Yeah.
Thanks, Teague.
Yeah.
Thank you very much.
Thanks for having me.
Great. Yes.
All right, everybody. And I actually have to go too, because I have to go teach a class in a minute. All right. So hopefully catch up with all of you later.
Brilliant. Well, thank you for giving such a great talk. And thank you everyone for participating and joining this webinar. And just remember, these webinars are happening on a regular basis. So keep an eye on the Bats Without Borders website or any social media to see what's happening next. Hey, thank you, everyone.
Bye, Riley.